All right, now let's go to BaseSpace. So this is in your browser window. The nice thing about all these software is it's on the web. So you don't really have to do any of that, computation. Your computer doesn't have to be, really, really powerful because most of the computations are happening away. Okay, is everybody on BaseSpace? You should be on Correlation Engine. Don't be on Cohort Analysis, which is another great program, but I can't show you because I don't have enough time. Everybody there? Okay, here's what we're going to do is now we're going to port that same gene list into this program. And I'll explain, after we get it in, I'm going to explain why this is so awesome. So name your study. I'm going to name it ABC versus GBC. I'm going to name it 7. Been doing a lot of these. Again, remember that import your data. You'll come up to this window. I am recording this. You can always go back and, figure this out, slow it down, stop it. You do have to listen to my voice while you do it. I'm sorry. Select appropriate data type. This is RNA expression. Click on that. Select experiment. These are peoples. Okay, now your next file, choose file. Again, it's the same file that we used in the last one, ABC versus GCB. SIG gene list, double click on that. Okay, now we're going to the next step. Again, just like Ingenuity, we have to tell it what columns we want it to use, what we want it to look at. We definitely want log 2 fold change, so in column B, as you go down here, select fold change log 2 mode. Okay. Down in column D, we want to put, this is the p-value. This is how we rank the genes. This is how significant they are. Keep going down here. Column I, it'll already be labeled gene probe set ID. I don't want that. So to get rid of that, go to choose custom. The only one that should say ID is column J. That's our ensemble. And we look down here, everything else looks to be custom is what we want. So basically, when I'm putting gene lists into most bioinformatics software programs like this, what it wants is it wants a unique identifier that it has a record of. Then it wants fold change, and then it wants a p-value. Those are the only 3 pieces of information you need. And you don't have to use genes, right? We could use proteins. We can use metabolic states. We can use anything. Okay. The top of your page should look green. If it doesn't look green, that's bad. Everybody look green? Green good. Green mean go. So go to the bottom and hit next step. So basically, we put in 747 identifiers. It was able to recognize most of them except 10, and that's pretty much normal. Some of these are just, isoforms, or some of the programs actually combine replicated genes. This is typically what you see. So let's go next step. Here we can put a tag in there. So say if we're working in a large group, if somebody, say at our university, wanted to look at our data, click on it here on enter tags, and what are we working on? Diffuse, and type in large, and then what will happen is diffuse large B-cell lymphoma will show up. It knows it already has these phenotypes in there, so click on that. That way, if you're working in a group or you're working with people, that you like, they can find your data. Trust me, working with people you like is a bonus. Okay, hit finish. Everybody's nodding yet. Yeah, I know. Congratulations. We have now imported this. And again, it's the same thing. They're basically going to ping it with their database. Go to home. I want to show you something about